Hello students, welcome to our online video tutorial class. I think you have seen the previous video, whatever I have made on the introductory part of the chapter, that is biological classification. There actually I have discussed about the different concepts of the kingdom, as well as I have discussed about the basic portion of the kingdom Monera. So I have decided that I will be just starting from the next, that means after the special features of kingdom Monera, whatever the points we have to study in this specific chapter under kingdom Monera. So let's see whatever the points we have to study at first. First of all, we have to discuss about the classification of the kingdom Monera based on the shapes. Next, we have to study about the classification of the Monera based on their other characteristic features. We have to discuss about the basic structural features of any specific bacterial cell, and at last we will be discussing about the other two type of bacterial cells, cyanobacteria, and next one, that is mycoplasma, and their characteristic features. So first we will start from the classification of kingdom Monera based on the shape. Specifically, if you study the shape of various bacterial cells, you are commonly can found four types of bacterial cells based on their shape. First one, that is a coccus bacteria. Second one, that is bacillus bacteria. Third one, that is a spidellum bacteria, and fourth one, that is a other shaped bacteria. Here, coccus bacteria means those bacterial cells which are round or spherical in shape. Bacillus bacteria means those bacterial cells which are rod shaped in structure. Spidellum means those specific bacterial cells which are spiral shaped. And fourth, other shaped means other any kind of the shape they can be shown. So first of all, that is the coccus bacterial cell. Specifically, this type of bacterial cell we have categorized them into various categories based on their number. How? If you can see that monococcus bacterial cell, that means we have to understand that there is a single coccus, a single spherical bacteria is present. Second one, that is a diplococcus. Diplococcus means when the two coccus bacteria, two round shaped or two spherical bacteria are just associated with each other, then it is commonly known as diplococcus. And third, tetracoccus. What is tetracoccus? Tetracoccus means four monococcus, that means four single coccus bacteria they have associated and formed the structure, that is the tetracoccus. Next, streptococcus. Streptococcus means when the coccus bacteria or spherical bacteria are present in a chain-like manner. Okay, they have actually made a chain-like manner, so for that reason that name has been given streptococcus. Next, Sarcini. What do you mean by Sarcini? Sarcini means here also the coccus bacteria are present in a chain-like manner, but the number of bacteria is limited. That is only 8. When the 8 bacterial cells are actually arranged in a chain-like manner, on that time that specific structure is commonly known as Sarcini. Next, Staphylococcus. Staphylococcus means this is the accumulation of coccus bacteria which are actually present like a bunch of grapes. So bunch of grapes like structure when it is shown by the coccus bacteria, and when the coccus bacteria are present in a group, on that time they are commonly known as Staphylococcus. So these are the different categories of coccus bacteria based on their arrangement, based on their number. Second one, you can see here the bacillus bacteria. Already I have told that bacillus bacteria means rod shaped bacteria. Here also, based on the configuration of various bacterial cells, how they are actually associated with each other, or how many bacterial cells, rod shaped bacterial cells are present, based on that four different categories can be found under bacillus bacterial category. So first one, monobacillus, it is a common sense, there is single rod shaped bacteria when it is present, on that time it is commonly known as monobacillus. Second one, diplobacillus. Diplobacillus means when the number of bacillus bacteria are 2 and they are just associated with each other side by side, on that time it is commonly known as diplobacillus bacteria. Third one, that is a streptobacillus bacteria, that means rod shaped bacterial cells are present but they are actually arranged end to end and they have made a chain like arrangement, then it is known as streptobacillus. Next, that is coccobacillus. Coccobacillus is such kind of bacillus bacteria which is oval shaped. Why oval shaped I am saying? Because you have seen the name coccobacillus. Coccobacillus means it is rod shaped as well as it is round shaped. That means overall it is actually bringing the shape of oval. So when the oval shaped bacteria are present, on that time they are commonly known as coccobacillus. Next, Spirillum bacterium. Spirillum bacterium is this such kind of bacteria which is spiral shaped. Spiral shaped can be directly, they can be comma-like. They are comma-shaped bacteria, like as an example, Vibrio cholerae, which is a positive organism of cholera. And second, spiral. That means they are actually spring-like structure showing bacteria. So they are spiral bacteria. So spiral and Vibrio, these two type of bacteria are coming under the Spirillum category. And the last category you can see here, other shaped. Other shaped means except this specific round shape or rod shape or Vibrio or spiral shaped, there are more other shaped shapes are also present. Let's see whatever the shapes. First one, palisade shaped bacteria. That means that these bacterial cells are actually looking like palisade parenchyma cells, elongated, and they are fleshy structure almost, and it is almost oval shaped structure. So palisade parenchyma like structure, when it is shown on that time it is known as palisade bacteria. Second one, pleomorphic. Pleomorphic bacteria means these kind of bacterial cells can show any kind of shape. There is no specificity of their shape is told, so they can show any kind of shape. That's why they are pleomorphic. Pleomorphic means multiple forms who can show. Hyphae shaped means we know what is hyphae. Hyphae is actually the thread-like structure or the fiber-like structure which are commonly found in case of fungal cells or in case of fungi. So like those thread-like structures, when the structure can be found in bacteria, on that time it is commonly known as hyphae. Next you can see here stalked bacteria. That means these bacteria are actually looking like a small stalk-like structure. So that's why their shape has been written stalked. So last one, trichome. What is trichome? Trichome means these specific bacterial cells are actually looking like the stem hair. On the surface of the stem, multicellular hairs are present, and those hairs are commonly known as trichome. So when the bacterial cells are those multicellular stem hair like structure showing, on that time they are commonly known as trichome. So here I have just entirely told you about the various shapes of the bacterial cell which can be existing in this world. Now I am going for the classification of kingdom Monera based on their morphological features, where they are actually available based on those. First of all, we have classified the entire kingdom Monera into two different categories or two different classes rather. First one, that is Archaea or Archibacteria. Second one, that is the Eubacteria. Archaea or Archibacteria means these kind of bacterial cells can survive in primitive environmental condition, where the environmental condition was very extreme. Normal organisms can't survive in that environmental condition, but few bacterial cells can survive in that extreme heat condition or extreme saline condition, extreme pH condition, so they can survive. So those bacterial cells are commonly known as Archaea or Archibacteria. Here one point always you have to keep it in your mind, that they have a special type of cell wall structure. Their cell wall composition is such kind of special so that they can survive in that specific extreme environmental condition. Okay, their cell wall is not made up of peptidoglycan. With the peptidoglycan, several other substances are also mixed and made the cell wall structure more complex so that they can survive in that specific extreme environmental condition. So those specific bacterial cells are mostly under this specific category. And another more important point, we have to keep it in our mind that these bacterial cells are mostly gram-negative bacterial cells. Okay, because extreme environmental condition, when they are surviving, obviously there is no sufficient quantity of oxygen present. So for that reason, they are mostly surviving in the anaerobic environment. This point also we have to keep it in our mind. Next one we can see here, Eubacteria, that is the true bacteria. This is the common bacteria, whatever we can see in our normal environment, in the air, in the soil, in the water, everywhere they are just loitering. So those kind of bacterial cells all are coming within the Eubacteria or the true bacteria. Now first of all I am going for the categorization of the Archibacteria or Archaea. Archaea or Archibacteria, that is a bacteria who are surviving in the extreme environmental condition. They are segregated into three different categories. Whatever the categories, first one, that is a methanogen or methanogenic bacteria. From the name we can understand here, these bacterial cells are actually methane forming bacteria. They are taking carbon, hydrogen, and they are forming methane. Okay, so they can be surviving in the anaerobic environmental condition. Obviously they are surviving in the stomach of the ruminating animals so that they can produce the methane, as well as they can be surviving in the marshy area, or they can survive in the swampy area where the methane can be produced. That means we have to keep it in our mind, these organisms or these kind of bacterial cells are fully chemoautotroph. They can just take the chemical substances like carbon and hydrogen from the environment and they are synthesizing methane. They can survive commonly in the anaerobic environmental conditions. So these specific bacterial cells are commonly known as methanogen. Example I have written, that means methanococcus. These are round shaped coccus bacteria, methane producing, that's why they name has been given methanococcus. Next, halophile. From the name you can understand they can survive in the extreme saline environmental condition where the salinity is very high. Other organisms can't survive. It's not possible for their survival. But few group of bacterial cells they can survive in that extreme saline condition. Why? Because they are taking those salts from the environment and they are just synthesizing their food for their survival. So automatically it is not hectic for them for surviving in the extreme saline environmental condition. So those kind of bacterial cells are also gram-negative bacterial cells, and they are also, keep it in your mind, they are surviving in the that anaerobic environmental condition because they are not getting oxygen at all. So example it has been told, that is halococcus. Here also the bacterial cell shape is round and they are showing affinity towards the saline environment. That's why their name has been given halococcus. Thermoacidophilic. Thermoacidophilic means when some bacterial cells are showing affinity towards extreme heat as well as extreme acidity of the environment, and they can survive in that extreme environmental condition. On that time, those bacterial cells are commonly known as thermoacidophilic bacteria or thermoacidophilic bacteria. Keep it in your mind, this kind of bacterial cells can survive more than 110 degrees Celsius temperature. Where the temperature is more than 110, there also they can survive, as well as where the temperature is pH 2, that means highly acidic, or below that also they can survive in a very well manner, okay, in a lavish manner they can survive in those kind of environment because they are anaerobic bacteria. They are taking those, they are surviving in that specific extreme heat as well as in the extreme acidic environment, because whatever the nutrients are required for their survival they are getting from that environment. That's why it's not impossible for their survival, it's getting possible for them. Example you can see thermoplasma, that is the name of the bacterial cell, has been given who can survive in this that extreme heat and extreme acidic environment. Next, I am just moving for the eubacteria. In the eubacteria, you can see here the first, it has been, the categorization it has been shown like that. First one, that is a normal bacterial cell. Second one, that is a cyanobacteria, which is commonly known as blue-green algae. Third one, it has been shown, that is a mycoplasma. So these three types of the bacterial cells are coming under the eubacteria category or true bacteria category, who are commonly, or which bacterial cells are commonly found in our environmental condition. So let's see about the bacterial cell. First of all, when we will be discussing about the bacteria, we have to know about their structure, their location. We all know they are surviving commonly in this our environment, in the tropic area, in the subtropic area. In these areas, temperature, humidity, everything is favorable for them, so that's why they can survive here. They are getting all kind of nutrients in this environment, so obviously it is possible for them to sustain their life. So whatever the structure is commonly present in case of this bacterial cell, that only we will see here. So first of all, I have segregated the bacterial structure into three portions. Whatever the three portions, first one the cellular envelope, that is the covering layers present in the bacterial cells. Second one, cytoplasmic substances, that means whatever the substances are present in their cytoplasm. And last, I have just written the point, that is the locomotory organ, that means by which organs the bacterial cells are just moving from one place to another place. So let's start the structure of the bacterial cell. First, when we will be discussing about the cellular envelope structure, the first point we have to keep it in our mind that first it is coming, that is the slime layer. Slime layer is the outermost very mucilaginous slippery substances layer, which is actually helping the bacterial cell to escape from any kind of environmental conditions. To escape from any kind of environmental condition that is actually, or any kind of predator, so that's why that slime layer is actually present. Second point we can see here, that is the capsule. That means just after the slime layer there is a protective membrane is present, that is very much hard, very much rigid, which is protecting the bacterial cell from any kind of environmental stresses. So that specific structure is commonly known as capsule. Third point you can see here, that is cell wall. What is cell wall? Cell wall is also a protective covering which is present just inside of the capsule, and this specific cell wall is commonly made up of peptidoglycan. Keep it in your mind, this specific portion is commonly made up of peptidoglycan. That is one kind of is a combination of polysaccharide and amino acid. And the next and the last covering, which is the innermost covering, that is commonly called as cell membrane or plasma membrane, and that is made up of phospholipid and protein. Normally those are the constituents of the plasma membrane, we all know. So these are the four coverings from outside to inside: slime layer, capsule, cell wall and cell membrane. So throughout these four different layers we can see in case of bacterial cell. Here one specific point we have to keep it in our mind, that in case of bacterial cells it has been found that the cell membrane is just protruding inward. Again I am saying the cell membrane is protruding inward and making several pouch-like structures, and those pouch-like structures, by protruding inward, those specific structures are commonly present for controlling the bacterial replication, bacterial respiration and those other activities. And keep it in your mind, these specific structures are commonly known as mesosome. These structures are actually present towards the cytoplasmic portion, and it has been made due to invagination or due to protrusion of the cell membrane or plasma membrane towards inward, that means towards cytoplasm. So that is called as mesosome, controlling the replication process, controlling the respiratory mechanism in case of bacterial cell. Coming to the next point, that is cytoplasmic substances. Whatever the substances are present in the cytoplasm of the bacteria, let's see whatever the substances can be seen. First one, that is the nucleoid. We all know that the nucleus is not well developed in case of bacterial cell. That is very primitive. So the nuclear membrane, nucleolus, this specific categorization in the nuclear structure, that cannot be seen. So for that reason, keep it in your mind, commonly whatever the structure can be found, that is known as nucleoid. Second point is that plasmid. Plasmid is that specific structure which can be also called as extra chromosomal DNA. And in this specific extra chromosomal DNA structure, keep it in your mind, several kind of virulent genes are present. Virulent gene means I am talking about those specific genes which are responsible for any kind of the diseases. So those kind of virulent genes are also present in this plasmid, and commonly it can be called as extra chromosomal DNA. Third one, inclusion bodies. What are the inclusion bodies? Inclusion bodies are those specific structures, those are vesicles or vacuole-like structures which are storing the excretory materials and before release. And when the time will come, these specific inclusion bodies will be fusing with the plasma membrane, and automatically the matters will be getting released outside. Food and gas vacuole. Whatever the food has been obtained by the bacterial cell, or whatever the respiratory gases have absorbed by the bacterial cell, those substances remain stored within these vacuoles, and from here, after digestion of the food and gas, they will be get distributed throughout the entire portion or entire regions of their bodies via diffusion process. And obviously ribosome should be present. That is the 70th category of ribosome, because that is a prokaryotic ribosome for the protein synthesis. So this is the entire structure of the cellular envelope and cytoplasmic substances. Except these two, there is also one more structure is present, that is the locomotory organ. What is locomotory organ? Keep it in your mind, in case of bacterial cells specifically, they need locomotion. They have to move from one place to another place. So for that reason, locomotory organ is also present. As an example, flagella is present. More than one flagella can be present in case of bacterial cell for their locomotion, as well as pili and fimbriae. These specific two structures are also present in case of bacterial cell. Commonly, these pili and fimbriae, both structures are responsible for sexual mode of reproduction, because during the time of sexual mode of reproduction, two bacterial cells, they are adhering with each other, but this pili and fimbriae, on that time they are obviously helping with that also, they are helping during this locomotory process or locomotory mechanism. So for that reason, pili and fimbriae, these two structures are also helping for the locomotion. Next thing is that here we can properly understood about the entire structure of the bacterial cell, that is starting from cellular envelope, cytoplasmic substances and locomotory organ. Next and the last 2 points whatever we have to discuss, that is the cyanobacterial cell and mycoplasma. What are these specific organisms? Except normal bacterial or eubacterial cells, there are some other kind of bacterial cells are present. As an example, first one, that is a cyanobacteria. This kind of bacterial cells are commonly called cyanobacteria due to presence of their blue pigment as well as green pigment, and they can be commonly called as blue-green algae. Now the point is, in case of cyanobacteria, whatever the features can be found, let's see. First one, they can survive in the polluted water. Okay, in the polluted water they are generally growing. In the fresh water they are not growing. Second one, they are commonly known as blue-green algae. They are showing algae-like shape. That's why they are commonly known as algae, but they are actually bacterial cells. Third, I have written a point, heterocyst. What is that? Heterocyst is a such kind of thread-like structure which is commonly found in case of this blue-green algae. Why it is present? To fix the atmospheric nitrogen within them, because they can also perform, cyanobacteria can also perform nitrogen fixation, like some free living nitrogen fixing bacteria or soil living nitrogen fixing bacteria. But keep it in your mind, this specific type of cyanobacteria, they can just fix the nitrogen within their body by the help of heterocyst. Heterocyst is that structure responsible for fixing nitrogen from the environment. They have phycocyanin and phycoerythrin, these two pigments, and due to presence of these two pigments, collective manner, these two pigments are commonly known as phycobilin. Keep it in your mind, due to presence of these two pigments, their color is mostly blue-green, because phycocyanin is generally blue color and phycoerythrin is red color, and with that chlorophyll is also present. So the red color is not shown outside, but the blue and green color, these two are just exposed towards outside, and that's why their color is blue-green. And these two pigments, again I am repeating, these two pigments are collectively known as phycobilin. Next, they are commonly gram-negative bacteria. What do you mean by gram-negative bacteria? Gram-negative bacteria is a such kind of bacteria or such category of bacteria who are not taking gram stain. Gram stain means when actually we are staining the bacterial cell for seeing under the microscope. Few bacterial cells, they are taking the crystal violet staining. Few bacterial cells are present, they are not taking the stain of the crystal violet. Crystal violet is the name of the stain. So who are taking the crystal violet staining, they are commonly known as gram-positive bacterial cell, and who are not taking, they are not showing that violet or purple color, they are showing the pink color. So those bacterial cells are coming under this gram-negative bacterial category. So cyanobacteria is a gram-negative bacteria because they will not take the crystal violet color. Next, they are commonly showing filamentous shape as well as they can be colonial also. And the next and the last important point about the cyanobacteria, that is they can store the starch, and that starch is a special kind of starch that is commonly known as cyanophycial starch. Cyanophycial means because they are containing that phycocyanin pigment, and for that reason they are showing this kind of starch, that is cyanophycial starch. What is the example of the cyanobacteria? Nostoc, Anamina, oscillatoria. These examples, I think this specific organism's name you have studied previously also. But here it is getting clear that these specific type of bacterial cells are coming under cyanobacteria category. Next and the last one, that is the under Monera, that is mycoplasma. What is mycoplasma? Keep it in your mind, mycoplasma is the smallest organism of the world. It is a bacterial cell, but it is the smallest organism of the world. It is even smaller than the virus. So for that reason, mycoplasma is the smallest organism of the world. Cell wall is absent in case of this kind of bacterial cell. In every bacterial cell, cell wall is present. In case of archaebacteria, special type of cell wall is present. In case of eubacteria, cell wall is there, which is made up of peptidoglycan. But keep it in your mind, in case of mycoplasma, that cell wall structure is fully absent. They are also gram-negative bacteria because they are not retaining the stain of crystal violet. They are showing the pink color under the microscope. That's why they are gram-negative bacteria. And another very important point, whatever we have to keep remember, that is mycoplasma can be also called as PPLO. Okay, what is PPLO? The full form is pleuropneumonia-like organism. It is written here, the pleuropneumonia-like organism. What is this? These organisms are looking like pneumonia causing bacteria. These specific organisms are looking like pneumonia causing bacteria, like a streptococcus pneumoniae, a diplococcus pneumoniae. So like them, they are actually looking, and they can cause the pneumonia like respiratory problem also. So for that reason, mycoplasma can be also called as PPLO. They are looking like pneumonia bacteria, that means like streptococcus pneumoniae, as well as they are causing the pneumonia like disease also in human being, respiratory problem. So that's why they are commonly known as PPLO. So this is the overall just difference, or you can rather say the characteristic features in case of cyanobacteria and mycoplasma. So we have just covered this entire portion, whatever I have written on the board. Again, I am just recapitulating in very brief. First of all, we have discussed about the type of bacterial cells based on their shape, that is coccus bacteria, bacillus bacteria, spirillum and other. Within coccus bacteria, we have seen various categories based on their number, based on their association. Under bacillus bacteria also, we have seen various categories based on their number, based on their association. Spirilla means specifically spiral-shaped bacteria, as well as Vibrio or comma-shaped bacteria coming under this category. And within other shape, calycian-shaped bacteria can come. Pleomorphic, that is multiple shape showing bacterial cell can come. Hyphae-shaped bacteria can come. Stalk-shaped bacteria can come, as well as stem hair or trichome-shaped bacteria can come also within this. So this is the totally classification of bacterial cell based on the shape. Next, I have classified, or the scientists have classified the bacterial cell or the Monera based on their characteristic features, where they can survive based on those. So which bacterial cells can survive in the extreme environmental condition? They are belonging from the category that is Archaea or Archibacteria. Another category of bacterial cells which are commonly known as true bacterial cells, who are surviving in this common environmental condition, the tropical area or subtropical area or whatever. Within Archaea, we can see there are three different categories of bacterial cells are coming. Mechanogen, that means methane producing bacterial cells. Halophile, that means that specific bacterial cells who can survive in the extreme saline environmental condition, and thermoacidophilic bacterial cells, that means who can survive in the extreme heat and extreme acidic environment. With those, we have also discussed about the eubacterial cells. Within that, we have talked about the structure of the common bacterial cell or true bacterial cell. In case of true bacterial cell structure, we can see three portions. One is cellular envelope, that is a protective covering. Second one, the various substances which is present or which are present within the cytoplasm. And the third one, that is the organs which are responsible or which are helping the bacterial cells for their locomotion from one place to another place. So let's see whatever the portion under the cellular envelope. We have seen slime layer, that is a slimy substance, mucilaginous substance, which is providing protection to the bacterial cell. Next, we have seen capsule, that is a hard protective covering just inside of the slime layer. Next, we have seen cell wall, that is also a wall-like rigid structure that is made up of commonly peptidoglycan, that is also protective in nature and very inside, that is the innermost layer, that is the cell membrane, which is commonly making the pouch-like structure by invaginating towards inward, and that is called as mesosome. That has also some physiological functions. Cytoplasmic substances, within that we have seen nucleoid, that is a ill-developed nucleus. There is no proper structure of the nucleus, and for that reason the nuclear material is just scattered in the cytoplasm, that is nuclear. With that we have seen the extrachromosomal DNA, that is the plasmid. Plasmid is containing the virulent genes, and those genes are responsible for causing various kind of diseases to different animal hosts. And next, inclusion bodies, we have seen various kind of small vesicle or vacuole-like structures which are containing various excretory materials. So those are commonly known as inclusion bodies. Next, we have talked about food and gas vacuole. In the food and gas vacuole, we know that food and gaseous substances remain stored, and according to requirement, they will be distributed to each and every part of the bacterial cell via diffusion process. Next, we have seen what is ribosome. In case of ribosome also, we know that the 70th ribosome, that is a prokaryotic ribosome, is present, and this ribosome is responsible for protein synthesis. At last, locomotory organ we have discussed. Within that, flagella, pili, fimbriae, these three different type of locomotory organs we have seen. Flagella is the most important locomotory organ. With that also two accessory locomotory organs are present. Those are pili and fimbriae. Last, we have discussed about cyanobacteria and mycoplasma. In case of cyanobacteria, we have told this is commonly known as blue-green algae. They are present in polluted water. They are gram-negative. They are containing heterocyst for nitrogen fixation. They are storing the starch, that is a cyanophycean starch. They can be filamentous. They can be colonial also. So these basic features are generally shown by the cyanobacterial cells. Examples have also written Norston Anamina oscillatoria. And last one, mycoplasma. That is the smallest organism of the world. They are even smaller from the virus. They are commonly known as PPLO because they are looking like that specific pneumonia causing bacteria, as well as they are causing the disease like pneumonia, and they are also gram-negative bacteria. And the most important feature about the mycoplasma, that is they are not having the cell wall. They don't have the cell wall like other bacterial cells. So this is the total portion whatever I have discussed in this video. I think everybody you have understood properly this specific portion whatever I have taught you today, and I think or I hope you have enjoyed this specific video a lot. So thank you everyone.